Peptomyc is a spin-off company of the Vall d'Hebron Institute of Oncology and of the Catalan Institute of Research and Advanced Studies in Barcelona. It aims at developing protein therapeutics for the treatment of cancer patients.
Hi, my name is Laura Soucek, and I'm co-founder and Chief Executive Officer of Peptomyc. Peptomyc is developing mini proteins against Myc.
Myc is essentially what allows cancer cells to proliferate, thrive, hide from the immune system, and, last but not least, resist to treatment. Notably, Myc is also present in normal proliferating cells, but it is not as essential for their survival as it is for cancer cells. That means that attacking Myc could selectively kill cancer cells without damaging normal tissues. Despite this indisputable therapeutic opportunity, there is no Myc inhibitor in the clinic yet, and the reason is that Myc has been so far considered an undruggable target. But we are about to change that paradigm. In order to function, Myc needs to dimerize with its partner MAX. When it's bound to MAX, Myc can assume the proper structure to bind DNA and direct gene transcription. So, we can inhibit Myc either disrupting its binding to MAX or to DNA. When I was still a student, I designed and characterized Omomyc, the best direct Myc inhibitor known to date. Omomyc is able to replace MAX as a partner for Myc, disrupting the Myc-MAX dimers and sequestering Myc away from DNA. In parallel, it can also form homodimers and the heterodimers with MAX that can bind DNA and switch off gene transcription. Hence, Omomyc has a double mechanism of action: direct sequestration and inactivation of the target, and competition for DNA binding. Over the years, Omomyc has been used as a transgene in multiple mouse models of cancer, demonstrating dramatic efficacy in different tissue contexts and against tumors driven by different oncogenic lesions, reinforcing the notion that Myc is the conduit for all of them. Amazingly, and against many preconceived notions, this did not cause any severe side effects in normal proliferating tissues that simply slowed down and did not suffer as a consequence of the treatment. This was published for the first time in Nature in 2008 and changed the dogma regarding Myc druggability. Now, in order for Omomyc to be translated into a pharmacologically useful tool, we needed something different from Omomyc in its transgenic form. Hence, Dr. Marie Beaulieu, co-founder and chief scientific officer of Peptomyc, started producing Omomyc as a recombinant protein in bacteria. This 10 kilodalton protein turned out to have unexpected cell-penetrating properties and was able to penetrate both the cellular and the nuclear membrane of cancer cells, causing their arrest and death. Importantly, this effect was limited to cancer cells and not normal cells. In fact, once Omomyc was inside the cancer cells, it would cause massive displacement of Myc from its target genes on the DNA. We calculated that this displacement occurs in more than 97% of Myc targets, and it is accompanied by epigenetic changes that could explain the long-lasting effect of Omomyc treatment. Myc displacement from DNA corresponds to a general transcriptional reprogramming characterized by the shutdown of the classical Myc fingerprint in all cancer cells. This is an absolutely on-target effect since it does not extend to other BHLHC proteins involved in cancer. Encouraged by these in vitro results, we proceeded with testing Omomyc's therapeutic effect in vivo. we first established that it could be administered locally to lung tumors through intranasal administration. The protein quickly reached the lung and accumulated in the lung tumors, where, once again, it caused Myc fingerprint shutdown and the reprogramming of the tumor microenvironment. The intranasal instillation of Omomyc in adenocarcinoma-bearing animals for 4 weeks resulted in a clear arrest of tumor growth and regression to adenomas or hyperplasia in more than half of the tumors. This was the result of reduced tumor proliferation, cell death, and recruitment of intratumoral CD3 positive cells, a finding that holds promise for future combination with immune therapy. However, the full potential of Omomyc was reached through its systemic administration. Indeed, weekly intravenous injection of the Omomyc mini-protein achieved therapeutic impact in a very difficult to treat non-small cell lung cancer model characterized by EGFR mutation, p53 mutation, PI3 kinase mutation, and resistance to erlotinib. Importantly, in these studies, Omomyc showed a half-life of almost 50 hours in the bloodstream. These data were published in 2019 in Science Translational Medicine. Since then, we have expanded our proof of concept to other types of cancers, such as breast cancer, in particular triple-negative breast cancer, and showed Omomyc efficacy in both preventing and treating metastases, besides primary tumors. More recently, a similar result has been obtained in colorectal cancer as well, even in the context of KRAS mutation. Peptomyc has now completed the industrial production of its first clinical batches and tested them in preclinical safety studies in rats and non-human primates. These studies have confirmed its safety and excellent PK and provided the basis for clinical trial application submission. The plan is to proceed to phase 1-2 clinical trials in Q1 2021, testing safety and efficacy of Omomyc in at least 3 different indications: KRAS mutant non-small cell lung cancer, triple-negative breast cancer, and RAS mutant colorectal cancer. Our team counts with a board of directors characterized by strong science, business acumen, and pharma out-licensing experience. It is also supported by an excellent scientific advisory board comprising eminent oncologists around the world. Peptomyc so far has raised more than 16 million euros in private investment and more than 5 million euros in public funds. This allows the company to initiate its phase 1-2 clinical trials. We are now looking for a round of investment of 25 million euros to be executed in Q2 2021 in order to lead our clinical plan until completion of phase 2A. If you'd like to learn more about Peptomyc and its disruptive new therapeutics for cancer treatment, do not hesitate to get in touch with us. Remember, Peptomyc.